Now let us differentiate between a prokaryotic cell and the eukaryotic cell. First of all, let's talk about the size of a prokaryotic cell. The size of the prokaryotic cell, is comparatively very much smaller as compared to a eukaryotic cell. If we talk about the size, then it ranges from 0.1 micrometer to 5.0 micrometer. And 1 micrometer, I know that you all know this, it is 10 raised to power minus 6 meters. So you can just make out how small is the size of the prokaryotic cell. But if we talk about the size of a eukaryotic cell, then the eukaryotic cell size is comparatively larger. It ranges from 5 micrometer to 100 micrometer. So you can again make out how can you compare the size of a prokaryotic cell with a eukaryotic cell. A prokaryotic cell is comparatively much smaller as compared to a eukaryotic cell. Now let's talk about the nucleus. In a prokaryotic cell, a well organized nucleus is absent. Means if we talk about the organization of the nucleus, we are talking about the nuclear membrane. Right? So that is absent in this case. So well organized nucleus is absent in case of a prokaryotic cell. But in case of a eukaryotic cell, we have a very well defined nucleus. It has a nuclear membrane, it has chromatin inside it, and it has nucleolus and nucleoplasm. So in case of a eukaryotic cell, we have a well organized nucleus that has a nuclear membrane, which also has chromatin, and it also has a nucleolus and nucleoplasm. So a eukaryotic cell has a well defined, well organized nucleus. Then the DNA is naked in a prokaryotic cell. What do we mean by naked DNA? Naked DNA means that in this case the DNA is not associated with histone proteins. What happens when there is arrangement of this DNA, or you can see that we have histone proteins, we have around 8 histone units, 4 on the front and 4 at the back. So around this histone protein units, the DNA coils itself like this, right? So this particular unit, which is formed of histone proteins and the coiled DNA, it is known as a nucleosome. So in a prokaryotic cell, this arrangement is not there, because the DNA is naked means it is not associated with histone proteins. But in case of a eukaryotic cell, the DNA is associated with histone proteins, it is not naked DNA, right? So that the arrangement that I have just explained you in brief, this is like this, means the histone proteins, they are associated with DNA. So the DNA is not naked in case of a eukaryotic cell. Then DNA lies freely in the cytoplasm. Obviously, we have discussed that a well organized nucleus is absent. So the DNA that is there, that is a naked DNA, it lies freely in the cytoplasm. But in this case, in the eukaryotic cell, we have a well defined, well organized nucleus. So the DNA is going to lie within this nucleus, right? Plus DNA is also present in plastids and mitochondria, right? Except for the nucleus, DNA is also present where? In the plastids, for example chloroplast, and in mitochondria. Okay, so you remember that some organelles, they also contain DNA, that is plastids. Example of such plastid is chloroplast, and then we have mitochondria, they both contain what? DNA. So in a eukaryotic cell, we have DNA within the nucleus and in the organelles like plastids and mitochondria. Then 70S type ribosomes are present in the prokaryotic cells. But in case of eukaryotic cells, we have generally 80S ribosomes, but 70S ribosome, they are present in plastids and mitochondria. So prokaryotic cell contains 70S type ribosomes, right? But in case of eukaryotic cells, we generally have 80S ribosomes, and 70S ribosomes are found in the organelles like plastids and mitochondria. Then membrane bound organelles are absent in case of a prokaryotic cell. So this is also a very important difference you can make out. Membrane bound organelles. What are these membrane bound organelles? We have ER, I'll write it here. ER is what? It is endoplasmic reticulum, right? Then we have mitochondria. Then we have Golgi apparatus, right? We have mitochondria, Golgi apparatus. We have lysosomes. These are the organelles that are membrane bound organelles, right? They have membranes. So these organelles, which are the membrane bound organelles, they are absent in case of prokaryotic cells, but these membrane bound organelles are present in case of eukaryotic cells. Remember this. Then in case of a prokaryotic cell, true or sap vacuoles, they are absent. Instead of these true vacuoles or sap vacuoles, we have gas vacuoles that are present in a prokaryotic cells, right? So we have gas vacuole instead of a true vacuole in case of prokaryotic cell. But the opposite is in case of a eukaryotic cell. In case of a eukaryotic cell, we have a true vacuole, a true sap vacuole, right, which is not there in case of a prokaryotic cell. Then in case of prokaryotic cell, as you all know that there is no sexual reproduction. So means meiosis, that is reductional division, is not going to take place. So there is no spindle apparatus that will be formed, as there is no meiosis, that is reductional division, which takes place in case of sexual reproduction. But in case of eukaryotic cells, sexual reproduction is there, meiosis, that is reductional division, is going to take place. So means that spindle apparatus will be formed in case of a eukaryotic cell. Then we have nucleoid equivalent to single chromosome is present. Now when we talk about the naked DNA that is present, which is lying in the cytoplasm, that naked DNA lying in the cytoplasm is also known as what? It is also known as nucleoid. And this nucleoid is equal to a single chromosome, which we also call as a prochromosome, right? So that naked DNA which lies freely in the cytoplasm is known as nucleoid, or you can also call it as prochromosome. Why we call it as prochromosome? Because this nucleoid is equivalent to single chromosome. But in case of eukaryotic cells, we have chromatin, and in chromatin we have many chromosomes, right? So in case of a eukaryotic cell's nucleus, we have chromatin material, and this chromatin material, consists of many chromosomes. It's not like equivalent to a single chromosome in case of prokaryotic cells. Then a prokaryotic cell has one envelope organization, means it is having one envelope. But in case of a eukaryotic cell, we have a two envelope organization. What do we have? We have a two envelope organization, right? So these are the points of differences that you can make out between a prokaryotic cell and a eukaryotic cell. Remember, first of all the size, then the nucleus, then what is more important is the membrane bound organelles that are not there in case of a prokaryotic cell, but these membrane bound organelles, they are there in case of a eukaryotic cell. So just try to understand what are the major differences between these two types of cells, and I hope after just tabulating these differences, the major differences that you need to know that I have listed here are very clear to you.